G-protein-coupled receptors, or GPCRs, are a large family of cell membrane receptors that play a crucial role in cellular communication. These receptors are linked to effectors, such as enzymes, channels, or carrier proteins, through GTP-activated proteins known as G-proteins. When an agonist binds to a GPCR, it triggers a cascade of intracellular events that lead to various physiological responses. GPCRs are involved in numerous bodily functions, making them a significant focus in pharmacology and medical research. All GPCRs share a common structural organization. Each receptor molecule consists of 7 alpha helical membrane-spanning segments made up of hydrophobic amino acids. These helices create 3 extracellular and 3 intracellular loops. The agonist binding site is located between the helices on the extracellular face, while the G-protein binding site is formed by cytosolic segments. The amino terminus of the receptor chain lies on the extracellular side, and the carboxy terminus is on the cytosolic side. This unique structure allows GPCRs to effectively transmit signals from the extracellular environment to the cell's interior. G-proteins are heterotrimeric, composed of alpha, beta, and gamma subunits. In their inactive state, GDP is bound to the alpha subunit. Upon activation by a GPCR, GDP is replaced by GTP, causing the alpha subunit to dissociate from the beta and gamma subunits. The activated alpha subunit, now carrying GTP, interacts with the effector, either activating or inhibiting it. The beta-gamma dimer can also activate receptor-operated potassium channels, inhibit voltage-gated calcium channels, and promote GPCR desensitization at higher activation rates. This intricate mechanism ensures precise cellular responses. Several types of G-proteins, distinguished by their alpha subunits, have been identified. The most important ones include Gs, which activates adenylyl cyclase and opens calcium channels, Gi, which inhibits adenylyl cyclase and opens potassium channels, Go, which inhibits calcium channels, and GQ, which activates phospholipase, C. Different receptors can utilize multiple G-proteins, a phenomenon known as agonist pleiotropy. For example, muscarinic M2 receptors couple with Gi and Go, while muscarinic M1 and M3 receptors couple with GQ. This versatility allows GPCRs to mediate a wide range of physiological processes. GPCRs function through 3 major effector pathways: the adenylyl cyclase pathway, the phospholipase C pathway, and the ion channel pathway. Each pathway involves different second messengers and leads to various cellular responses. The adenylyl cyclase pathway results in the accumulation of cyclic AMP, CAMP, which activates protein kinase A, PKA. The phospholipase C pathway generates inositol trisphosphate, IP3, and diacylglycerol, DAG, leading to calcium release and protein kinase, C, PKC activation. These pathways are crucial for regulating numerous physiological functions, from heart rate to hormone secretion. Activation of adenylyl cyclase by G's proteins leads to the production of CAMP, a second messenger. CAMP primarily functions through CAMP-dependent protein kinase, or PKA, which phosphorylates various enzymes, ion channels, transporters, and transcription factors. This results in increased heart contractility, smooth muscle relaxation, glycogenolysis, lipolysis, and modulation of hormone synthesis. Additionally, CAMP directly opens cyclic nucleotide-gated calcium channels in the heart, brain, and kidney. The action of CAMP is terminated by phosphodiesterases, which hydrolyze it to 5-AMP, ensuring precise control of cellular responses. The phospholipase C pathway is activated by GQ proteins. Phospholipase C hydrolyzes phosphatidylinositol 4, 5-bisphosphate, PIP2, to produce IP3 and DAG. IP3 diffuses into the cytosol and mobilizes calcium from the endoplasmic reticulum, while DAG remains in the membrane and activates PKC with the help of calcium. PKC then phosphorylates various intracellular proteins, leading to diverse physiological responses. The cytosolic concentration of calcium is tightly regulated by specific pumps to ensure proper signaling. This pathway plays a vital role in processes such as muscle contraction, secretion, and cell growth.